Hello world! I've got something pretty exciting to show you today. Eat leeks, beat SARS-CoV-2. I hope you'll take a leak with us. www.leakaweek.org. First, the disclaimer. Everything you see in here is going to be taken directly from existing scientific research. I have zero credentials whatsoever that are relevant to this. You are encouraged to verify the information yourself. The only thing I claim to have is Google Food, the ability to find information on the internet, which I am presenting to you today in this video. Now, that said, I think if you eat leeks, you can beat SARS-CoV-2. Everything I'm about to show you can be summarized in these two comic strips, and I hope you will take a leak. I apologize in advance for the length of this video. Immunology, as it turns out, is a little bit complex, but we're going to work through it today, and I'll show you what I learned. When this pandemic first started, I wanted to look for foods that might help my body fight the coronavirus in the event that I was exposed. Now, what I found was this paper, Plant Lectins are potent inhibitors of coronaviruses. And from this paper, everything else followed. In this paper, here's its abstract. They described antiviral activity of plant lectins against severe acute respiratory syndrome coronavirus, SARS-CoV. Now, this is the 2002 version, which is the predecessor, a previous version of SARS-CoV-2 that's circulating around the globe today. They said the strongest anti-coronavirus activity was found amongst Mannose-binding lectins. And two targets of antiviral intervention were identified with respect to SARS coronavirus. They found it early, roughly around viral attachment when the virus tries to infect you, and at the end when the virus tries to replicate. This is a pretty interesting paper, so I looked more deeply into it, and you found this result. All mannose-binding lectins, except for those from garlic, had anti-coronavirus properties. Now, most interesting is the most potent lectin against SARS coronavirus is the mannose-binding one from leek. So if we just take the two green underlined sentences together, what we see is leek mannose-binding lectins demonstrate strong anti-coronavirus activity against SARS coronavirus, SARS-CoV. Again, 2002 version. Knowing nothing about what these are, I wanted to look up mannose-binding lectins and what it does, what it is. So I found this, the role of mannose-binding lectin in innate immunity. Here's its abstract. The innate immune system, which includes mannose-binding lectin, recognizes a broad range of molecular patterns on infectious agents. Pretty cool. They think MBL plays an important first-line host defense role against infectious agents. Now that's pretty cool. So what do these MBLs do? Well, this paper, Mannose-binding lectin and the balance between immune protection and complication, goes into that in more detail. A pivotal link between the innate immune system and other functions is mannose-binding lectin, a pattern recognition molecule. What they found was the protective mechanisms of MBL include efficient opsonophagocytic killing of pathogens and activation of the lectin pathway. Now, I didn't know what opsonophagocytic means, so I Googled that. It means phagocytosis initiated by an opsonin. You'll see those words come back up again later, so we'll come back to that. But what's more interesting is lack of MBL, right? In MBL deficient hosts, that tends to lead to systemic infection, right? And uncontrolled inflammation due to cytokine storms. Now remember, at the beginning of this pandemic, a lot of cytokine storms were being seen in patients. People didn't understand why. But here we actually get a hint about that. what we see is MBL protects us by killing pathogens, and having low levels of MBL leads to cytokine storms. All right. So what else does this paper say? Well, successful innate immune protection is achieved in 2 steps. First, identify the target, and second, eliminate it. Pretty simple. How do you activate it? Well, you activate the innate immune system by using soluble pattern recognition molecules, such as MBL, which normally your liver produces and is circulating throughout your body and your blood. They also found MBL in the lung, even when you're healthy. And so what this research concludes is MBL, as a pattern recognition molecule of your innate immune system, provides surveillance both locally and at possible points of contact with the external environment and systemically. So throughout your body and at the entrances and exits. And in this manner, MBL can contribute both to immunity from pathogens and a bunch of other stuff. So let's summarize this. Innate immune protection has 2 steps. 1, identify targets. 2, eliminate targets. MBL activates the innate immune system. And MBL contributes to immunity from pathogens by acting like security guards on patrols and at entrances to your body, right? Like the lungs. So let's do a quick recap of what we've seen so far. Leek Mannose-binding lectins demonstrate strong anti-coronavirus activity against SARS coronavirus, SARS-CoV. Again, that's a 2002 version. MBL identifies targets for our innate immune system to eliminate. And MBL circulates in our bloodstream and is in our lungs patrolling for pathogens. And having low MBL may lead to more infections and cytokine storms. Now that we understand MBL and its activity against SARS coronavirus a little bit, let's talk more about the immune systems, right? This innate immune system. Turns out we have 2 immune systems and something called the complement system. One of the immune systems we discuss most nowadays is the adaptive immune system. And the reason for this is because vaccines teach our body to create antibodies to fight specific pathogens. Now, our adaptive immune system requires training. It needs to learn how to fight these pathogens, and that training can take up to 14 days. Once trained, it typically responds to an infection in 4 to 7 days. Now, vaccines are great. We have a lot of really good examples of good vaccines. Flu, measles, right, MMR, chickenpox, tetanus, DTaP, and polio. These are all really good vaccines. But they all train the adaptive immune system. The immune system that we're talking about today is the innate immune system. These are just separate. The innate immune system is not specific against a particular pathogen. It's actually broad spectrum against lots of pathogens. And this innate immune system is always active, 24 hours a day, 7 days a week, 365 days a year, and it's our first line of defense against infections. So for example, skin tries to keep germs out of your body all of the time. 2 other parts of the innate immune system, which we're going to talk about a little more today, is Mannose-binding lectin and phagocytes, right? And this is what this video wants to focus on, the innate immune system. Now, part of our innate immune system is the complement system, which you activate to destroy pathogens. And as you can see by this paper, complement is a functional bridge between the innate and adaptive immune responses that allows an integrated host defense to pathogens. So basically, the complement system is a bridge between the innate and the adaptive immune systems in order to have your whole immune system as together fighting pathogens. Now, I think of the complement system as an all-purpose recycler, and you'll see that in a little bit. So the question here is, how do you activate the complement system to destroy pathogens? Here's a picture from the same paper, activation pathways for the complement system. You'll see on the left, under the yellowish orange, This is the classical pathway. The classical pathway is how your adaptive immune system activates the complement system, right? And you'll see antibody here and antibody here. So antibodies initiate the classical pathway. The innate immune system activates the lectin pathway, which is in the middle in green. And here you'll see recognition by lectins and MBL, right? So MBL can bind to common carbohydrate PAMPs, P-A-M-P, on ... some viruses and parasites. PAMPs is short for pathogen-associated molecular patterns. You'll see this term come up a little bit more later. This paper also has another picture of that kind of illustrates what happens. So MBL and antibodies, when they recognize a pathogen, like a microbe, so let's say this MBL and this antibody, when they start recognizing a pathogen like this microbe, the MBL and antibodies attach themselves to it. And when they attach themselves to it, a macrophage, this phagocytic cell, comes and eats the microbe. So now what's important here is all three activation pathways, that's the classical pathway, the lectin pathway, and the alternative pathway, all three of them initiate the opsonization pathway of complement. So that's this part on the bottom right here, right? And that's the tagging and clearance of foreign bodies. So the innate immune system, when a lectin recognizes a pathogen-associated molecular pattern on a pathogen, like when MBL might recognize SARS coronavirus, it kicks off a stream of events, right? And that results in opsonization. Now, opsonization is an immune process which uses opsonins to tag pathogens for elimination by phagocytes. So, in essence, MBL tags pathogens for your immune system. If you look at, let's say, Wikipedia, which has a generally more English, understandable description of things, you can see MBL. It's a lectin that's instrumental in innate immunity as an opsonin and via the lectin pathway. If you look up opsonin, opsonins act as tags to label things in the body that should be phagocytosed, which is a fancy word for eaten by phagocytes that specialize in phagocytosis, cellular eating. If you look up phagocytosis, phagocytosis is a mechanism used to remove pathogens by digesting them, right? So very literally, MBL tags pathogens for your innate immune system to eat and destroy, which brings us back to the title of this video. Eat leeks, beat SARS-CoV-2, but more specifically, your innate immune system will eat SARS-CoV-2. So, quick recap, let's add that to the list of things we know, right? MBL tags pathogens for your innate immune system to eat and destroy. Now, so far we've looked at MBL against SARS coronavirus, the 2002 predecessor to SARS-CoV-2. To figure out if leek MBL would work against SARS-CoV-2, let's first look at how MBL recognizes SARS coronavirus. And we found this paper. A single glycosylation site of severe acute respiratory syndrome coronavirus spike facilitates inhibition by Mannose-binding lectin. All right, that's kind of cool. One place that MBL binds to inhibits SARS coronavirus. And here's its abstract. Again, Mannose-binding lectin is a serum protein that plays an important role in host defense as an opsonin and activating the complement system. Now, serum proteins, kind of a new word here, we look it up, and it basically just means a part of your blood, right? Now, the key part of this paper, though, a single glycosylation site, N330, was critical for interactions between MBL and SARS-S. S is the spike. They also go on and say their results identified a single glycosylation site, N330, on spike glycoprotein as target for interactions by MBL and SARS coronavirus. So basically, MBL binds to SARS-CoV at N330, right? And we saw leek MBL binds to SARS coronavirus. So what does that mean? Well, this paper has a very nice picture. They basically mapped the spike of the SARS coronavirus. Now, if you remember, the SARS coronavirus is this virus that has a lot of little spikes on the end of it. So this image that it's showing you is basically one of those spikes, right? There's a top view looking down at that spike and a side view from the side. Now, the cool thing about this picture is they labeled N330. They found it, right? And this N330 is where leek MBL binds to. And it's at the top of the spike, which is very accessible, right? It's kind of like the outside of a soccer ball. But how does that relate to SARS-CoV-2? Well, we found this paper, which says 2019 nCoV is a new coronavirus very similar to SARS-CoV. And they say that N330 corresponds to N343 in the spike of 2019 nCoV and is a conserved glycosylation site. Now, it turns out 2019 nCoV was the interim name for the virus before it was named SARS-CoV-2. So, N330 corresponds to N343 on SARS-CoV-2. And what does conserved mean? If we look up conserved, it means retain unchanged. Now that's pretty cool because this one paper says N330 of SARS coronavirus from 2002 is retained unchanged and corresponds to N343 in SARS-CoV-2. So let's look at N343. Again, here's another paper which has a picture of the spike of SARS-CoV-2. And they labeled N343. Again, at the very top of the spike, highly accessible. So let's put these two viruses together. What we see is SARS coronavirus on the left, SARS-CoV-2 on the right, N330 at the top of the spike. N330 is retained unchanged, corresponding to N343 in SARS-CoV-2. Now, that's basically the same picture. And we know leek MBL binds to SARS coronavirus at N330, and since N330 corresponds to N343, leek MBL should bind to SARS-CoV-2 at N343. All right, let's recap that. MBL should recognize SARS-CoV-2 just as it recognizes SARS coronavirus because the key binding site is unchanged, right? N330 and N343. And MBL is your first line of defense against viruses and bacteria. So think of it like this. If I can recognize you in one photo, I could probably recognize you in another photo, right? That's kind of how this leek MBL works. So let's look a little bit more at MBL in general and how it recognizes pathogens. I found this paper, Lectins, innate immune pattern recognition molecules. MBL is a lectin. Now, they have this picture which displays kind of an image of MBL. And you'll see here this red trimer portion. That's the head. So the head of MBL is, there's a picture of that with a three-dimensional trimeric human MBL. And that's the head of this lectin. That's got the carbohydrate recognition domain, CRD, right? This carbohydrate recognition domain recognizes patterns on pathogens. So this is the business end, the recognition end. And this paper goes on to say a broad carbohydrate specificity is required by lectins to recognize and bind a large repertoire of pathogen-associated molecular patterns, PAMPs. And such broad specificity is achieved by an open and flexible binding pocket within the carbohydrate recognition domains. So here's the heads, this orange and red part on the three heads. That's the recognition part, and you'll see that it's got this blue springy part in the middle. That's the neck. Now the neck allows the heads to have some flexibility and movement so that it's not fixed, right? You can have this flexible binding from the MBL. And they say that a wider range of binding specificity has been reported for MBL. So MBL's very broad range recognizer because it's got this flexible head structure in this triangular pattern. And let's look at the tail. Now the tail of this MBL, the collagen region, activates the immune system, right? And we see here complement activation and opsonization and phagocytosis, right? Same things we've been looking at before. So let's put this all together. We have MBL, which has a head with a carbohydrate recognition domain, and we know that leek MBL recognizes SARS coronavirus at N330, and we know N330 corresponds to N343 in SARS-CoV-2, and it's conserved, which means it's retained unchanged. So leek MBL almost certainly will bind to SARS-CoV-2 at N343. So let's add that to our summary. MBL almost certainly recognizes SARS-CoV-2 as it recognizes SARS-CoV. And we know this because MBL is a very broad recognizer of pathogen-associated molecular patterns. And it's the same pattern, right? It's unchanged. So there's one more thing, variants, right? Now, here's a new paper. It's a preprint, admittedly. It's still new. It's not peer-reviewed yet. Too new for that, actually. So what did they find? They found that all 22 glycosylation sites of SARS-CoV-2 are highly conserved amongst major variants. And they're using lectins. And there's hoping that these lectins can provide pan-variant therapeutic interventions. Right? In the paper, all 22 N-glycan sites of the spike remain highly conserved amongst all the predominant SARS-CoV-2 variants. And this indicates a selection pressure to preserve these sites, meaning these sites are, for some reason, important to the virus. We'll come back to this. But again, because these sites are conserved, they're emphasizing the potential for pan-variant therapeutic interventions. Now, what does pan in front of the word mean? Pan means all or every or all-inclusive, right? So all major SARS-CoV-2 variants have the same lectin binding sites on their spike. And yes, this includes N343. Lectins that target these sites could treat every variant. So let's add that to our body of knowledge. Leek MBL very likely binds to every SARS-CoV-2 variant. Now we saw earlier that the virus tries to preserve these sites. Is N343 important? So let's look at this paper, where they basically tested a bunch of different mutations in the lab. And one of their tests found that deleting N343 reduced infectivity. Specifically, when they deleted N343, they reduced infectivity by 20x, right? And they suspect that the N343 is critical to somehow binding of the receptor or maintaining the shape of this virus. In any case, the 20x reduction in infectivity says it's important. So N343 is critical to the SARS-CoV-2 infection process, but it's also a weak spot. The virus needs N343 to infect us, but it's also recognized by MBL in our immune system. which means targeting N343 with MBL is aiming for a critical weakness. So we can take a look at a little more deeply into N343 with this paper. Again, a little bit on the new side, so it's still a preprint. It's not peer-reviewed yet. But what we see here is that the virus, the spike, has to transition from this down state to an up state. It basically has to change shape when it tries to infect us. And N343 plays a role in opening the spike to infect us, right? They identified a glycan at position N343 that participates in the opening mechanism. So what they found was there's a bunch of residues. Most of them we don't care about. The one we do is N343. These are conserved between SARS coronavirus, the 2002 version, and SARS-CoV-2 spikes because they help the spike open, part of the infection process. And the emerging mutant SARS-CoV-2 strains, long list, let me summarize it real quick. There's alpha, there's beta, there's gamma. This is still new. They may not have gotten to delta, lambda, and all the later ones. But at least in the emerging SARS-CoV-2 strains, they do not contain mutants in the residues identified here. And again, N343 is in this list because N343 is needed to help the spike open. So the virus needs N343 to open the spike during an infection, but N343 can be recognized by MBL in our immune system. N343 is conserved not just from SARS-CoV in 2002, but across many of the SARS-CoV-2 variants that we see today, which means targeting N343 is aiming for a critical weakness that SARS coronavirus needs to be infectious, right? In essence, it's an Achilles heel, right? N343 is the Achilles heel of the SARS coronavirus, and leek MBL can target it. If you like Star Wars, N343 is the exhaust port of the Death Star. So let's add this to our list of knowledge. Leek MBL targets a critical weak spot at N343 and N330 by recognizing the Achilles heel of SARS coronaviruses. Now, leek is just a vegetable. Can you simply eat it? Obviously, with the title of this video, Eat Leeks, probably the answer is yes. Kind of a spoiler there. Here's some science behind it. So, lectins are in many of our foods, right? It's almost impossible to avoid these lectins. Many of these lectins are stable against heat denaturation, which is good because it means they won't be destroyed by cooking. Some of them will be, but not all of them. And we're constantly eating these. Intake of lectins from diets and feeds can be significant. Recent studies have reported that lectins show diverse effects, including immunomodulating and antiviral activities. Now, immunomodulating basically means a substance that affects the functioning of the immune system. We can also see that many lectins resist digestion, survive your gut, and enter the circulation intact, maintaining full biological activity, which means they can do what they normally do. Now, this is all very complicated science, but it really boils down to one thing, which is vegetable lectins power our immune system. Eat your vegetables. They're good for you. They've been silently helping your immune system fight infections, right? And do we have evidence of lectins in action with our immune system? Well, turns out, yes. There's a Dr. Gundry. He's written The Plant Paradox. Now, he finds, and this is from the back cover and inside flap of his book, I took this off Amazon. He's found that lectins, because they're in a lot of our foods, some of his patients are suffering from autoimmune issues. And an autoimmune disease is an illness that occurs when the body's tissues are attacked by its own immune system. So in essence, Dr. Gundry has found some people who are eating some lectins are finding that those lectins are causing their immune system to target the person, which is bad, right? And these lectins are everywhere. But what this really means, though, is it's not so much a paradox, but rather it's an allergy, right? An allergy is a damaging immune response by the body to a particular food. And if you're eating a food that has a lectin that's targeting you, your immune system at you, you essentially have not a paradox anymore, but a plant allergy. And again, there's many, many different lectins. Some of them are bad, some of them are good. So here's a crude Venn diagram. There are some bad lectins, right? And not every lectin is bad for everybody. Some people are allergic to peanuts, others are not, right? Mannose-binding lectins tend to be very good, right? And there's other lectins, which we're skipping for today. So what does Dr. Gundry say about leeks? Well, he has this leek soup, and if you Google it, you'll find that he says it's lectin-free, right? Wait, lectin-free, yes. Now that's confusing, because we've just been talking about Mannose-binding lectin and leek. So how do you reconcile this? Well, leeks definitely have a Mannose-binding lectin, and yet Dr. Gundry says his leek soup is lectin-free. Well, I see two possibilities. The first is that maybe the leek lectins are destroyed by high heat and prolonged boiling. Possibly, maybe. Except Dr. Gundry actually recommends eating leeks. Leeks are on his yes list for foods to eat, right? If you click on that link and you go to it, if you look under foods to eat, leeks are there under yes. So I don't think the lectins are being destroyed in leek. What I think is happening is Dr. Gundry is looking for dangerous lectins, lectins that cause problems to people. Leek lectins are dangerous lectin-free. They don't cause problems to people, so these leek lectins are not on his radar because they don't cause problems. Now, if you remember, there's a lot of lectins out there, so you can think about them on a spectrum. There's definitely bad lectins, right? Ricin's a terrible lectin. Kidney beans are pretty bad for you, but if you cook them, it's not so bad. On the other end of the spectrum are good lectins, right? Black elderberry, stinging nettle, garlic all have good lectins, and leeks have good lectins. There's a lot of other lectins that are just also in the middle, right? And some of these may be good for some people, bad for others, right? So for example, take peanuts. Peanuts are fine for most people, but some people are terribly allergic, right? And some people may just be mildly allergic to some foods. So each individual is going to be reacting differently to different lectins. So what does this mean? It means avoid eating foods you're allergic to, and especially if you're allergic to a particular lectin, because a lectin allergy describes the autoimmune reaction where the lectins are targeting your immune system at you. That's bad. Avoid those foods. On the other hand, eat as much of the healthy foods that you're not allergic to as you want, right? Because they don't cause any problems for you and they help your body fight pathogens. But more importantly, you can choose food for their beneficial lectins to help you fight certain infectious pathogens, right? Because every vegetable lectin recognizes a different kind of pathogen. And eating a wide variety of vegetables gives you a wider spectrum of protection. Now, we happen to know leeks recognize SARS coronaviruses. So eating leeks will help you beat SARS-CoV-2. And specifically, it will help your immune system eat SARS-CoV-2. Now, you might ask, what if you're wrong? Well, in the worst case, you ate a vegetable. You ate a leek, maybe you don't like it, who knows? If you're allergic to or don't want to eat leeks, don't eat it. That's probably the worst case that can happen here. In the best case, though, your 24/7, 365, always active innate immune system helps you kill SARS-CoV-2 when you eat leeks. That's a pretty huge upside. So how much leek should I eat? Well, our website is leekaweek.org, so a leek a week. We have a bunch of recipes online to help you get started. Subscribe and thumbs up to learn more. Our next video is going to go through the math for why a leek a week. And again, everything you just saw in this video can be summarized into these two comic panels. Recipes are on our website, Take a Leek a week. And if you're interested in helping or contributing towards a study, please email us at contact@leekaweek.org. Thank you.